Assalamu alaikum dear students, how are you? Fine. Today we are going to discuss the new chapter that is enzymes. Now first of all, what are enzymes and what is their function? We have to discuss this. Now actually enzymes they are biological catalysts. What is the meaning of biological catalysts? They speed up reactions in our body. For example, we can take an example that we have eaten a paratha and egg in breakfast. So it will take several hours to digest. But with the action of enzymes in mouth, in stomach, and in intestine, this whole breakfast is digested in very less time, maybe an hour or two. So we can understand with this example that enzymes they speed up of the chemical reactions. हमारी बॉडी में enzymes कैसे होते हैं like protease, amylase, these are all enzymes and they are made up of proteins. They are all proteins. Okay, so this is again this speed up the rate of reaction without themselves being used up. Now let's move ahead. A chemical reaction in our body can be to convert proteins into amino acids or anything. So what happens that all these enzymes they work at their specific pH and their optimum temperature. And all the enzymes end with the word ase, like protease, lipase, amylase. Now, for example, what enzyme does in our body? There is a substrate. Substrate is the substance on which the enzyme act, and products are the reaction substances that are produced after the reaction of enzyme with substrate. Now, let's take an example. For example, we have protein as a substrate. Protein as a substrate. On protein, this protein might be present in our egg that we have consumed in the breakfast. Protease enzyme will work on this protein substrate and will convert it into products like amino acids. So these amino acids are products. Okay, now how these enzymes work? Enzymes they have a specific binding site or a catalytic site. So what happens that the substrate bind on these substrate like for example protein will bind on this binding site of protease enzyme and then products will be formed like amino acids. We can see this in this reaction. Now for example, this is an example of enzyme protease and this is substrate protein. So enzyme substrate complex means protease enzyme plus protein substrate complex has formed. Now protease has become free, it can be reused and amino acids are the products. So this is the reaction. This reaction is also explained by lock and key hypothesis. Now what lock and key hypothesis says? Lock and key hypothesis says that as a specific key can open a specific lock, similarly specific enzyme will work on specific substrate. Like lipase enzyme will only work on lipids, and similarly amylase enzyme will only work on starch. And we have discussed above that protease enzyme will only work on protein. So that is why these enzymes are specific in nature. And like above, now you can see that this is an enzyme molecule. Protease will bind with protein, lipase will bind with lipid, and amylase will bind with starch. So this is their specific action. Now you can see that the substrates, they are binding with the enzyme molecule. The reaction is being carried out, and here the products are formed. Again, this is another reaction. And in anabolic reactions, making of substances are occurring, and catabolic breaking of substances are occurring. Now you can see that this is the substrate, it is binding with the binding side or active side of the enzyme. Enzyme substrate complex is formed, and here again the products is formed. So this is lock and key hypothesis. The breakout of this reaction is protease will never bind with a lipid, or lipase will never bind with a starch molecule. Moreover, Lock-and-Key hypothesis also suggests that the structure of the enzyme is a rigid structure. Means a rigid structure means no change occur in the structure of the enzyme during reaction. No change occurs in enzyme shape during a reaction. So it is fixed, rigid structure. So this is the second point of lock and key hypothesis. Now, factors affecting the rate of enzymatic action. Okay, now we have to look at this graph. This is temperature on x-axis, and this is rate of reaction of enzyme on y-axis. Now you can see that at 0 degree centigrade, the enzymatic action was 0. But for every 10 degree rise in temperature, 10, 20, 30, 37, for every 10 degrees centigrade, the enzymatic action is being doubled. For example, at this is enzyme and this is substrate. For example, at 10 degrees Celsius, if one enzyme was working on two substrates, so at 20 degrees Celsius, one enzyme will be working on 4 substrates. Similarly, at 30 degrees Celsius, one enzyme will be working on 8 substrates. And then at 37 degrees Celsius, which is the maximum, then the double of 8 is 16. So means at 37 degrees Celsius, one enzyme is working on 16 substrates. So 37 degrees Celsius is also called as the optimum temperature at which the enzymatic action is maximum. After this, you can see that the enzyme action is decreasing. Okay, from here why it was increasing? Because when the temperature was increasing, more temperature, more collisions between enzyme and substrate. When there were more collisions between enzyme and substrates, so ultimately more products were formed. And when the temperature was decreasing, now less coolants and thus slow rate of reaction until at 60 degrees Celsius reaction stops. Why reaction stops? Because at 60 degrees Celsius enzyme is denatured. For example, if this is the shape of a normal enzyme, then this enzyme is denatured. Now it cannot bind to any substrate and it is wasted. So for every 10-degree rise in temperature, the rate of enzymatic action is doubled until 37 to 40 degrees Celsius, which is the optimum temperature. Now after 37 degrees Celsius, rate of reaction begins to slow down and 60 degrees Celsius enzyme is denatured, shape of active site changes and no longer now the substrate can bind to active site of the enzyme. The next level is pH. Every enzyme works best at its optimum pH. And optimum pH is different for different enzymes. Like for example, for amylase works best at 7 pH, but protease in stomach it works best at 1.5 to 2 pH, and lipase that is in the intestine it works best at 8 to 9 pH. So this amylase is in mouth, it works best at 7, and this in stomach protease it works best at 1.5 to 2, and lipase in intestine it works best at 8 to 9 pH. So all the enzymes will work at their optimum pH. If the enzymes have to work at pH other than their optimum pH, so enzymes will be denatured and shape of the active site will be changed and no substrate can now further bind to the enzyme and enzyme has become useless. Now you can see that pepsin is working best at 2 pH. At 3 pH, this pepsin is denatured. Similarly, most enzymes like amylase, they work best at 6 to 7 pH. Other than their pH, they will denatured. Similarly, lipase, it is working best at around 8 pH and at 11, lipase will also be denatured. So, other than their optimum pH and other than their optimum temperature, enzyme will be denatured. Now we have to prove this experimentally that enzyme work best at their optimum temperature. So we added, first of all, we took a test tube. In the test tube, we added egg white, and this egg white is pure protein. And in this, we added protease enzyme. So one test tube like this we kept in crushed ice. Other test tube we kept at room temperature 25 degrees Celsius. This was test tube 1. This was test tube 2. This was test tube 3 at 40 degrees Celsius. Test tube 4 at 60 degrees Celsius. Test tube 5 at 80 degrees Celsius. And so we will see that in crushed ice, no working. So albumin egg white will remain as it is. At room temperature, some of the albumin will be digested, but since it is not the optimum temperature also, some of the albumin will still remain. At 40 degrees Celsius, all the albumin will be digested. At 60 degrees Celsius, enzyme will be digested. So egg white albumin will remain there. In 80 degrees Celsius, again egg white albumin will remain there. Why? Because in both these cases, enzymes they are denatured. So this is about same we can perform with the pH. Like if we protease works best at 1.5 to 2 pH. So at 1.5 to 2 pH, egg white will be digested. At 3, 4, 5, 6, 7, 8, enzyme will be denatured and albumin will not be digested. So this is all about enzymes, and you can properly read from these notes also. This is another example that is proving the working of the enzyme. That, for example, uh, we know that amylase enzyme digests iodine. So we took two petri dishes and we poured iodine on them. In one iodine and plus starch, iodine plus starch. In one petri dish uh, we added starch, and the other petri dish we also added starch, and then we added amylase in one, and the other we added some other enzyme. So we can see that where amylase is added, it will be digested and the whole will become bigger, and in the other no digestion has occurred, hence prove that only specific enzyme works on specific substrate. Thank you very much. Assalamu alaikum. Take care of yourself.